Since it is such a common reproductive pattern in living things, both animals and plants, we're going to talk about cell interactions in terms of sperm and egg cells, males and females, genders, at least male-female genders. And talk in terms of chemotaxis, the sperm finding an egg cell. The situations vary. Perhaps the sperm has traveled down a pollen tube where the tube itself is primarily finding where the egg cells are, and then it's released pretty close. In some cases, it's sperm are released into the environment and they have to basically track chemical concentrations down a gradient to find where the egg cells are. The egg cells are releasing signaling chemicals. In internal systems, land animals, where the sperm are released into the body of the female, very often the chemotaxis of the sperm changes depending on where it is in relation to the egg cells. Is that when it actually gets where the egg cells are, it needs to be able to try to get into one. When it's on its way up the female system to try to get to where the egg cells are, and it hits the surface, it shouldn't react to the surface like it needs to get into it. It should bounce off and keep swimming. And so its chemotaxis changes its behavior depending on, I believe it's calcium ion concentrations. It's something where when it gets close to the egg cells and it hits the surface, it embeds and tries to get inside. How does it do that? Number one, in a lot of animals, this is an oversimplification. There's not a single cell floating naked inside the system. It's actually got a bunch of ovarian cells around it, like a layer or sometimes a couple of layers. And the sperm have to get through those in order to get to the egg cell membrane. In the front tip of the sperm is an endosome, is a vesicle called an acrosome, which is basically tip body is in Latin. And it releases enzymes that allow the sperm to work its way between the cells and open a hole to get its nucleus into the egg cell. That is the first real step of fertilization. But in most cases, there's a whole bunch of sperm trying to do that at the same time. Open a hole, pop the nucleus in. Now the nucleus should be the only part of the sperm that gets into the egg cell. Because sperm are mobile, mobility is energy using, energy requires mitochondria, and mitochondria have their own DNA. And it may be different DNA than what's in the egg cell. So the sperm mitochondria and the egg cell mitochondria may be genetically different. In most cases, if the sperm mitochondria got in and mixed with the egg cell mitochondria and all the subsequent cells were a mixture, probably not a big deal. But there are conditions where this does not work properly. Sperm mitochondria get into the egg cell, become a mixture inside the embryo and then the cells of the embryo, because their mitochondria are reproducing as the cells make more cells. Sometimes they're not compatible. Sometimes they battle it out metabolically, and that can cause conditions. In some cases, for many years, is that mitochondrial DNA was the gold standard of evolutionary DNA analysis, molecular clocks and stuff, because it was always assumed that like my mitochondria came from my mother's egg cell and not my father's sperm. It's biology. Nothing's always. And they've since found that sometimes it's the father's mitochondria. Sometimes it's a mixture of mitochondria. Most of the time, vast majority of the time, it's coming down a maternal line, egg cell to egg cell to egg cell. So I don't get to pass my mitochondria onto my offspring, but what I have is what my mother had. So that's one thing in the fertilization process that needs to kind of work right. Just the nucleus, and just one. Now, the egg cell itself is got detectors on the surface to detect the opening of a hole big enough for a sperm nucleus to come through. And the response of the egg cell is to immediately change the surface of the membrane. Because remember, the sperm are using an enzyme. Enzymes are a molecule-to-molecule fit in order to do something. Well, if the molecule on the egg cell changes shape, this doesn't fit anymore. And that is a way to shut down all the other sperm that are trying to open holes at that particular moment. Does it always work? Just one nucleus gets in? No. Is that there's now a condition called polyspermy, multiple sperm. More than one sperm nucleus gets inside, and now you've got the potential for, instead of one set of chromosomes from the egg cell and one from a sperm, there's now 2 from sperm or 3 from sperm. And as we'll deal with when we get to the genetics chapter, multiple copies of genes beyond the 2 you're supposed to have can be dangerous for a lot of genes, very dangerous for embryo development. And an embryo that develops with 3 chromosomes in animals likely to die pretty quickly. So what keeps this from happening? In some animals, nothing. Is that they produce so many egg cells and so many offspring that they can let the occasional ones that get polyspermed to just die. They're going to crank out hundreds of thousands of offspring anyway, so what the heck. In ones that do respond, there are a number of different ways that responses happen. In some, one nucleus is sequestered, the other ones are put into vesicles and taken to the surface and rejected, dumped out. In some of them, they're surrounded, they're put into food vacuoles and they're digested. Basically, everything but one is somehow dealt with, leaving just the one sperm nucleus to fuse with the one egg cell nucleus. So again, I'm not writing a lot of this stuff down, but it's a video. You can back up and write stuff down as we go. In plant cells, in plants, I mentioned that in animals, extra sets of chromosomes can be, are deadly for the most part. In plants, a whole extra set of genes can change the features of the plant to the point where it can actually produce a new species. What's happened in crop development, artificial selection over thousands and thousands of generations, is a lot of times you get what you're looking for in a polyspermed offspring. Example. Corn, modern agricultural corn, is a descendant of teosinte, a wild plant. Looks more like, the heads look more like wheat than they look like corn ears. No, but they have little kernels in them. If you genetically compare them, they do have, there's a lot of homology in the genes, except teosinte, the wild version, has 2 sets of chromosomes. Modern corn has 8 sets of chromosomes. So over the course of breeding for certain traits, variants with extra sets of chromosomes were selected and continued to be bred with until the modern version has many, many, many extra sets of all of its genes, because plants are okay with that, is that plants are okay with a lot of weird stuff genetically. I want to back up a little bit. Another thing that happens in animals. I have absolutely no idea how this works in plants. I just realized I've never run into this described anywhere, so I'm not even going to try to say this. But in animals, the egg cells are produced through meiosis, right? 2 divisions. And that division is a complex process. But making a working egg cell by putting a lot of food and the material needed to get the embryo going is also a very complicated process. So very typically in animals, those don't both happen at the same time. So very, very often, while the egg cell is accumulating food, it isn't really an egg cell. It has not gone through meiosis 1 yet. In mammals and in some animals that do ovulation, the egg cell develops inside the ovary and is released in the direction of the oviduct, and the sperm are going to be waiting in the oviduct. There's a small window of time where it's no longer getting food because it's disconnected from the food supply, and it's not out where the sperm are. Get to that in a minute. And it will do its first meiotic division at that time. That's what happens in mammals, happens in us, is that what's released from the ovaries, again, not an egg cell yet, has gone through the first meiotic division and is now in the oviduct, still not an egg cell, has produced one polar body, and the egg cell itself still has double-stranded chromosomes. But once it's in the oviduct, now it has another complicated job. It needs to let one sperm in and keep all the other ones out. It can't be dividing on the surface while it's trying to do this, because remember, polar bodies are produced right on the surface. So it doesn't. Once one sperm nucleus has gotten in and the other ones have been shut out, maybe it deals with extra ones at that point, maybe it waits, but it's going to wait, because once it's closed the membrane off to more penetration, invasion, however you want to put it, then it does its second meiotic division. And now it's actually a legitimate egg cell nucleus, one set of single-strand chromosomes. And then the sperm nucleus and the egg cell nucleus can do what's called fusion. They fuse together, and now you have two sets of chromosomes, the normal eukaryote complement in animals. In some animals, no meiosis happens until the sperm is let into the cell, because there's no safe time to be doing it because there's other stuff going on. In those animals, very often, there's not a small window of opportunity between where the egg cell is released and where the sperm are waiting for it. A lot of times it's just, and there's sperm all over the place. But once you have fertilization, fusion, you now have a zygote. A zygote is the first cell of an embryo. So it has all of the genes, all the alleles, all of the variation that it's going to have. But it needs to have more cells. So it divides, and 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 it divides. Okay, I'm just looking at my notes are a little bit out of order from what I want. I hopefully have a video of this process, the cell division, and some of the early processes of embryo development. There was just a new video put out that I'm trying to link into the book. Right now my servers are all messed up, so hopefully it'll be there when you go look at it. But you wind up with a ball of cells that does hollow out. Now, does it actually form a ball? Maybe. In most land vertebrates, this embryo is sitting on top of a very large yolk in an egg. And because the yolk is round, the embryo, as it divides and divides, instead of forming a ball, it actually forms a little disc because it's sitting on top of this big thing. And rather than be like an iceberg with half of it inside the yolk, it kind of sits on the surface like sort of like a frisbee. So that we always show it as a ball, but it isn't necessarily a ball. This blastula, that's what the hollow ball is called. A dent forms in it, and now you're going to have what's called gastrulation. So you have this ball, and you get this dent, and cells start to migrate in and form a second layer inside. So this is going to come in and form a second layer inside. Now you're getting layers of cells that are eventually going to do different things. What stays on the outside is called the ectoderm. It's going to form things you would expect, stuff on the outside. It's going to form skin. It's one thing that's a little surprising because you don't necessarily realize it starts on the outside. It forms nervous system. Nervous system starts as a surface, that set of cells that winds up on the inside eventually. And considering that your nervous system is primarily there to react to outside stimuli, that may explain why it's, ancestrally an outer development. And then, other things that your skin does. In some animals who have their skeletons or shells on the outside, those are derived from ectoderm. This inner layer is the endoderm. It's going to be, this space is going to connect to the outside. It'll, in most animals, connect at another end to the outside and be a long tube. So here's a huge surprise. It's going to be the digestive system. And there's going to be things that grow off of that. Gills are going to grow off of that. Lungs are going to grow off of that. Some glands are going to grow off of that. A lot of glands of your digestive system are a combination of ectoderm and endoderm, but things associated with the digestive system are at least partly endoderm. There's two, we're going to get into animals now. There's two main branches of animal relationships. There's the protostomes and the deuterostomes. Is that we are the deuterostomes. The protostomes are the mollusks and the arthropods primarily, and a lot of little groups. The deuterostomes are the echinoderms and us, the chordates, and a lot of other little groups. And there's a lot of differences between them. So be prepared to make yourself a 2-column table and leave yourself some room because there's a few things that are going to happen later that are going to go on that list as well. But here's a few things to start with. When these cells start to divide, this is a deuterostome. How do I know that? Because the cells are all the same size. Protostomes, the cells divide unevenly, and you wind up with a little cell and a big cell as the cells divide. And when you look at the ball that eventually forms, the little cells tend to get pushed to the outside in a spiral pattern. Cell division, another word for cell division is cleavage. This is called spiral cleavage. And in deuterostomes, when you watch the cells divide, hopefully you can watch that video because you'll see it. You get a division line and then a division line and then division lines, and they look like radii in a circle, and that's called radial cleavage. That's one of the primary differences. Another thing, in protostomes, these are starting to get programmed very, very early. So very early in embryo development, these cells already have their eventual jobs programmed in. They have some flexibility, and there are some stem cells that have some additional flexibility, but there's much more of a sealed fate to the cells very early in an embryo, whereas in deuterostomes, the cells remain very, very flexible for a really long time. This is why if something happens that a blob of cells separates out, even from a somewhat later embryo, it can form an identical twin. It can form a complete second animal. In deuterostomes, you're not going to get that. You're not going to get identical twin deuterostomes because the cells, you're going to lose cells that were programmed to do some part of the original animal. They're not going to be there. The original animal's in trouble. They're not going to form a whole animal. It's a bad thing. Now we get to where the name comes from. This initial dent, this initial opening of the digestive system in the protostomes is going to be the mouth. In the deuterostomes is, at least in theory, going to be the anal opening. In a lot of deuterostomes, it actually is the opening to the umbilical cord, but Eventually, there's an anal opening as well that grows down from the end. It also turns out that the mesoderm, the middle layer, that's going to fill in the middle, forms differently in the two groups. Okay. I always have to remind myself of this. Deuterostomes, you get a growth here. In protostomes, you get growths out, little blebs off of the endoderm that form the mesoderm. I don't have a good picture of this, so I'm going to say I'm pretty sure that's the way it works, but I may be wrong about that. Now, obviously, we've had this before. Hox genes are starting to kick in. Hox genes that are eventually going to determine what the front end is going to be and what the right side is going to be and where this part of the nervous system is going to form and where that part of the nervous system is going to form. Remember, Hox genes are very basic layout pattern inducers. And in fact, in most advanced animals that tend to be metameric, where they're somewhat segmented, is that you've got a head end and a tail end and then body sections, is that the Hox genes are often lined up on chromosomes in the order of the body sections. So it's a holdover from a distant past. Okay, I'm just looking to see. Talk a little bit about, I'll come back to the nervous system. The nervous system forms on the surface and then either sinks or either pulls the skin on each side or above it and then sinks that way, or it sinks in. It depends on, protostomes and deuterostomes have different setups for their nervous systems. I don't want to really get into that. One's dorsal, one's ventral, one's hollow tube, one's not, one's double, one's single. We're not getting into that. We already did that in the first semester. But now you're getting into the cells start to interact, right? That's what the chapter's about, cell interactions. The Hox genes start to produce different chemistries in the cells, and they start to release different chemicals into the area. And so they talk to each other. And a lot of development where the cells start to do different things, obviously, is them expressing some of the genes on the DNA, but not other genes on the DNA. This is called differentiation. What are we going to call it when they start to be different? We'll call it differentiation. And so muscle cells are going to be using muscle genes, and skin cells are going to be using skin cell genes. And there's going to be some genes that are active all over the place. Making more DNA. If they, there's all sorts of things are going to happen. All the cells are going to do, but a lot of cells are going to do different things. And what you eventually get is then the cells start to move around. If you look at the video, you'll see a lot of times you're seeing blood cells, circulating. The blood system forms very, very early in vertebrate embryos. The heart starts to beat incredibly early because you need that circulation to move materials around and to get waste materials away from the cells and get nutrients and oxygen from the yolk and the surface into the cells. So if you watch the video, I remember going, oh, there's a lot of stuff moving around. I realized, okay, that's blood cells in blood vessels for the most part. But there is migration as well, is that your nervous system are all outgrowths of a central stalk where the nerves reach out to all the different places that you have nerves in your body. Some cells stick together. It's called adhesion, and you form clumps because remember, your organs are a bunch of different cells working together, and something needs to hold them together. A lot of cells communicate, right? Your nerve cells are branching out from your spinal cord trying to get down to your fingers. Well, as it gets there, it'll go past clusters of cells that will go, okay, you need to go that way. That's the end of the blob that's going to be the arm and the fingers. And then as it goes down, just go, okay, I already sent one that way, you need to go this way. And eventually, so you get branches that go to all the fingers and not a whole bunch of branches in just one finger. In some cases, an organ will grow, and there's a point at which you need it to stop. And it will hit something nearby that will go, okay, you're as big as you need to be right now. Stop adding cells. It's called contact inhibition. And that's something that's occurring. I think I'm going to stop there. Pick this up in the next video.